Students, I want to meet you once again to discuss the lesson sex determination. And in this one, we have to know more about the Y chromosome and the male development, the development of male characters. Now here I given the structure of Y chromosome. Now if you are taking the Y chromosome, detailed analysis has been made regarding the genes and the different regions of the chromosome and their potential functions. Now, in Y chromosome, normally it is acrocentric. The Y chromosome is acrocentric. That means the centromere is present towards one end of the chromosome. Just actually as a result, we have one short arm, one long arm. Now in the case of this one, this Y chromosome has certain regions. These regions are homologous counterpart on the X chromosome. Those regions which are homologous to the X chromosome are called pseudo-autosomal regions, PAR. Pseudo-autosomal regions or pseudo-autosomal region, PR. So these regions are homologous to the X chromosome. Normally, the X-linked genes and Y-linked genes are not homologous. There is no homologous counterpart. But we have, if you are going detailed study, the Y chromosome has pseudo-autosomal regions called PAR present at the tips of the Y chromosome. You see the shaded region, that is a PAR, pseudo-autosomal regions. They are nothing but homologous counterpart on the X chromosome. So this region normally pairs with X chromosome, then recombine, synapses. During meiosis, this region just normally pairs, synapse, and then actually recombine. During meiosis, actually exchange of what is called chromosomal materials occur between X and Y chromosomes in these two regions. That means once again crossing over occurs between these two regions. That is what we have, the pseudo-autosomal regions and the region in the X chromosome, both are homologous. That's why it's called as actually pseudo-autosomal regions, similar to autosomes but not originally, but have a homologous, that is counterpart with the X chromosome. That is why it's called pseudo-autosomal region, present at the tips of the Y chromosome. Now it constitutes about 5% of the total length of the Y chromosome. The remaining 95%, what we can say, NRYH, the remaining 95%, NRYH. So pseudo-autosomal regions form only 5%. The remaining that forms what is called 95% called NRYH. That is non-combined region of Y chromosome. Non-combined region of Y chromosome. What I mentioned here, what is called MSY. This is what is called, otherwise called NRY. Non-combining region of what is called actually Y chromosome. So we can say also NRY here, non-combining region. That means it is not homologous with the X chromosome. There is no homologous counterpart in the X chromosome. So it is not forming a pair with the X chromosome. Does not what's called recombine. Does not undergo crossing over with the counterpart in the X chromosome. That's why it's called non-combined. That is region of Y chromosome NRY. It is not homologous to what is called X chromosome. Now, this NRY region is functionally divided into two components. One, euchromatin, another one, heterochromatin. One, euchromatin, another one, heterochromatin. Now, the euchromatin forms the functional region and the functional genes region. And the heterochromatin forms the non-functional genes region. What is the difference between euchromatin and heterochromatin? If you are taking interphase nucleus, suppose this is an interphase nucleus, we have the chromatin fibers. At certain regions, there is a condensed region of chromatin. The condensed region of chromatin is called heterochromatin. The condensed region of chromatin is called heterochromatin. And this one, the diffuse region of chromatin is called what is known as euchromatin. Otherwise, the darkly stained region of chromatin is a heterochromatin. The lightly stained region of chromatin is called euchromatin. Now when stain is applied, it is darkly stained, it is lightly stained. Now some more differences, darkly stained, lightly stained. This is a condensed region of chromatin, this is non-condensed or diffuse region of chromatin. It contains normally more DNA, it contains more DNA, it contains less DNA. Though it contains less DNA, it is genetically active. But the heterochromatin, though it contains more DNA, it is genetically inactive. So the Y chromosome is normally genetically inactive because it contains more heterochromatin. The Y chromosome is genetically inactive because it contains more heterochromatin. So you can see the euchromatin, heterochromatin only during the interphase nucleus. Now this is the interphase nucleus. These are all the chromatin fibers. The chromatin differentiated into euchromatin and heterochromatin. Now, there is a difference between euchromatin and heterochromatin. Y chromosome is rich in heterochromatin that renders the Y chromosome inactive. X chromosome contains actually more euchromatin. That's why it is normally genetically active. Coming back to this one. So 95% we have actually, that is inaugurated, non-combining region of Y chromosome. It is differentiated in the functional genes area called euchromatin, non-functional gene areas called heterochromatin. Now within the euchromatin, there is a region. Now what I mentioned is SR1. This SR1, the expansion for that one, it is a region which decides the sex of male. So sex determining region of Y chromosome. SR1 is called sex determining region of Y chromosome. It is present within the euchromatin and is towards one end, just behind what is called pseudoautosomal region. Now, this region, SRV region, is responsible for the development of the male characters. Its product is called testis development factor. Its product. The region is coding for testis development factor. Testis development factor. This is a product of this, what is called the region SRY. It is present in adult male, responsible for the development of what is called the testis and associated male characters. This is about what we call the Y chromosome and what is its role in the development of male character. So, it encodes for testis development factor, that is normally by what is called SRY region. That's region coding for the TDF. So anyway, now the Y chromosome is genetically inactive because of the concentration of more heterochromatin. But within euchromatin, we have the region, SRY region, responsible for the development, responsible for the production of a factor, what's called testis development factor. That factor is present in adult male testis, responsible for the development of masculine characters. Close. Students, we will pass on to the next part in sex determination. What is dosage compensation? That is a God body. Now, if you are taking what is called female, they have 2 doses of X-linked genes as they have 2 X chromosomes. In contrast to this one, the males have only 1 dose of X-linked genes as they have only 1 X chromosome. This creates a problem, what is called dosage problem, inequality in number of genes. So, 2 doses of genes, 1 dose of genes, that creates what we call dosage problem. This dosage problem can be compensated by what is called a structured bond body. So that method is called dosage compensation. How to compensate the dosage? We cannot add X chromosome to the male, but we can inactivate one of the X chromosomes in female, so the dosage compensation occurs. So while we are inactivating one X chromosome, now the females have one active chromosome, the males also have one active chromosome. This phenomenon is called dosage compensation, also called Lyon's hypothesis. Lyon's hypothesis. This is what is called dosage compensation, inactivation of one of the X chromosomes. Now, this Bohr body was first reported by Bohr, hence the name Bohr, and another person, Bertrand. These two persons observed the Bohr body in the female, actually cat, the nerve cells of female cat, because there only you see that one become the female's two X chromosomes. So Bohr and Bertrand observed the Bohr body. What is Bohr body? It is nothing but the condensed region of chromatin, what we can see already, heterochromatin. Condensed region of chromatin, otherwise called heterochromatin. And as it is present, the X chromosome is heterochromatin, is called sex chromatin. Now, Bohr body is a sex chromatin or heterochromatin present in one of the X chromosomes of female, not in male. Now, the presence of sex chromatin inactivates that X chromosome. So, now the females have only one X chromosome, similar to what is called one active X chromosome of male. This is called dosage compensation. So, what is Lyon's hypothesis? Inactivation of one of the X chromosomes by the presence of inactivation of one of the X chromosomes by the presence of Bohr body or sex chromatin or heterochromatin is called Lyon's hypothesis. So Bohr body inactivates the X chromosome where it is present. So both males and females have actually same dosage of genes, and that is called dosage compensation on Lyon's hypothesis as it was proposed by Lyon. Lyon only just actually proposed. The inactivation is because of the presence of the Bohr body. Now, it is being used in Olympic Games, whether the female participant is a true female or not. Now, the Bohr body follows what is called N-1 rule. The Bohr body, number of Bohr bodies, you can see, number of Bohr bodies is equal to number of Bohr bodies is equal to N minus 1, where N is equal to number of X chromosomes. Number of X chromosomes. Suppose we are taking a normal female, they have 2 X chromosomes. Here we have 2X minus 1, so they have 1 Bohr body. So if 1 Bohr body is present, then that individual is called actually a female. If you are taking a normal male, so they have only 1X minus 1, no Bohr body. So the females are called positive to Bohr body. Positive to Bohr body. The males are called negative to Bohr body. Suppose in the case of one syndrome, what is called Klinefelter syndrome. Klinefelter syndrome. We will be studying later sex chromosomal anomalies. There you have, this is the pattern of chromosome. 44 autosomes XXY. So extra actually X chromosome is present. Normally this is a normal male, generally we can say. So they have 44 autosomes. Because of the presence of one extra X chromosome, they develop feminine characters. So in the case of Klinefelter syndrome 2, we have just actually 1. Actually N minus 4, 2X minus 1, we are getting just 1. Though you have 1, that is Bohr body is present, they are not considered as actually true female because of the presence of that is Y chromosome. So in Olympic Games, the female participant is a true participant female or not. They are actually calculating this N minus 1 rule, and accordingly a true female has only 1 Bohr body. Though we have in this one, they are going for the karyotyping for displaying for the syndrome. Another method, whether they have what's called extra X chromosome or not, for that we are using karyotyping. But just if you have only one body, that female is allowed to participate in preliminary games. This is the test used in preliminary games, an entrance examination question. Okay, that's about dosage compensation and body. Let's talk about another method of sex determination seen in some insects. What is called haploidy diploidy type of sex determination. It is observed in the case of honeybees. That is, in the case of one species, I am giving the example Apis mellifera. That is one honeybee, where the males are haploid, the females are diploid. The males have 16 chromosomes and the females have 32 chromosomes, indicating haploid diploid number of chromosomes. This was first observed by Charles Bonnet. Now, when the male is undergoing actually gametogenesis, forming the gametes or the spores. Now, as the somatic cells are haploid, the gametes, namely the spores, also have n number of chromosomes. The spores also have n number of chromosomes. So, the nature of cell division during the formation of male gametes likely to be mitosis. In mitosis only, the parental cell and what we have, the gametes have same number of chromosomes. Unlike the male, if you are taking the females are diploid and they undergo what is called a process of cell division during the gamete formation to form haploid eggs. So the nature of cell division here is meiosis. So the male gametes are formed by a process of mitosis and the female gametes are formed by a process of meiosis. So here n number of gametes are formed. That is the eggs. Suppose if fertilization occurs, if the female gamete or the egg is fertilized by what is called actually the sperm, then we have diploid number is restored if fertilization occurs. So some of the eggs undergo fertilization to form a diploid female. So that is a result of offspring with a diploid number. And some of the eggs undergo parthenogenesis. They do not undergo fertilization process. They directly develop into what is called male individual. That is the reason why the males have n number of chromosomes. So the females are developed by means of sexual reproduction. That is called biparental sexual reproduction. The males are developed by parthenogenesis. Such a method of parthenogenetic development of haploid males, from unfertilized egg is called arenotoky. The parthenogenetic development of haploid males from unfertilized egg is called arenotoky. This is a phenomenon. The parthenogenetic development of haploid males, that is from unfertilized egg, is called arenotoky. Now, if you are taking the male, they do not have father. That is one statement is given in CBS book. The males resulted do not have father, but they have a grandfather. The meaning for that one, the female, the mother, is formed by the union of male and female gametes, possibly from a father and then a mother. So, this male is developed directly from what is called the mother without father. That's why we can say the resultant male has no father, but they have grandfather. So that's about what is called haploid and diploid type of sex determination. And this one, what is the importance of haploid and diploid type of sex determination? So here, there is one phenomenon we have to discuss about this. What is called kin selection. What is called kin selection. What is kin selection? So based on this what is called haploid and diploid type, we have to relate this kin selection. This concept was given by Charles Darwin. This concept was given by Charles Darwin. So it is an evolutionary strategy. Kin selection is an evolutionary strategy in which one organism favors the reproductive success of its relative. Favors the reproductive success of its relative. I give the example. Even at the expense of its survival and reproduction. So kin selection is an evolutionary strategy in which an organism favors the reproductive success of its relatives, that means its own species, at the expense of its survival and reproduction. This concept is called kin selection. Now, this kin selection normally is responsible for the evolution of sociality, responsible for the evolution of sociality. This is significance. What is sociality? That is society, a group of individuals living together in a cooperative manner with a division of labor. A group of individuals living together actually in a cooperative manner with the division of labor. Now it is observed in the case of honeybees. In the case of honeybees, we have three different types in the colony. One is queen, is normally diploid. Another one, worker. This is also diploid. This is nothing but a female, sterile female. You can say sterile female, not able to reproduce. Sterile female. Another one, the drone. This is what's called again just actually n number. This is sterile male. Sterile. Sorry, we cannot say sterile male, so we can say simply this drone. So queen diploid, worker diploid, but it is a sterile female, and that one is drone, that is normally fertile in nature. Fertile. And the queen is also fertile. Now this is what's called evolution of sociality. A group of individuals living together in a cooperative manner where there is a division of labor. Each one is performing a particular function. Now the queen bee is responsible for the laying of eggs after fertilization. Now what is the role of worker or sterile females? They are responsible, they are helping raising the egg. Raising the egg of queen. This is number one. They are responsible for raising the eggs of queen. And also contribute to the reproductive success. Contribute to the reproductive success of queen. I mentioned already what is kin selection. There is an individual favors the reproductive success of another organism that is nothing but its own relatives, even at the expense of its survival reproduction. Here, the worker bee is responsible for raising the eggs of queen and so contribute to the reproductive success of the queen. So this is one organism favors the reproductive success of another organism, here the queen. So the female sterile worker favors the reproductive success of what's called queen, even at the expense of its own survival or reproduction. So it doesn't undergo reproduction, but favors the development of what is called actually the queen by raising the eggs of queen and also contribute to that is a reproductive success of queen. So reproductive success of queen depends on mainly the sterile female. This is called kin selection. So it is responsible for the evolution of sociality. That's why I mentioned it is an evolutionary strategy favoring the reproductive success of one organism, here the queen, by another organism, namely what is called worker, even at the expense of its own survival reproduction. That means it doesn't undergo reproduction, but indirectly involved just in reproductive process. This is what is called actually kin selection as proposed by Charles Darwin, an evolutionary strategy responsible for the evolution of sociality, that is living together in a cooperative manner with the division of labor.